Very good evening, students. We start at 16:07 or so. We just updated our timetable today. Only 15, 13 people have joined so far. I want to teach today about microscopy mainly, not much. Shall we start now? Okay. So basically, microscope is used to magnify the smaller objects. So at the moment, I will be telling you about basics of microscope, that how it works, uh, it see to see the naked eye from to see the difficult things from the naked eyes. So that's the basics of your microscope here. That you might be doing from your biology class in your class 10th, when you have a CBSE board and you are given this microscope to tell you that which part is which. So I'm gonna brushing up your old memories here in order to go to advanced microscopy first. So so here you have your body tube, you have ocular lens, you have nose piece, you have objective, you have stage clips, you have a diaphragm, then you have a light, base, fine adjustments, coarse adjustments, stage, arm, ocular lens. So all these parts together make your microscope. So there are three types of microscope. That is simple microscope, compound microscope, electron microscope. So simple microscope has only one lens, whereas the compound has two sets of lenses. Okay, and it can magnify 100 to 200 times larger than they really are. And the electron microscope has objective up to 300,000 times, and they do not use lenses but use electrons to enlarge the image. So ocular to see from the eyepiece, whereas the coarse adjustment is a large knob on the microscope that bring the project into focus. Then fine adjustment, smaller knob on the microscope that brings the image into the focus. Then you have arm, hand on the back. Then you have a body tube, and then you have a nose piece. So all these together makes your microscope. Then high power objective lens is there. Then low power objective lens at the on the side, and the middle power. So it has 40x, 3x, and 10 to 20x. So three different magnifications of your um, objective lens. You have a stage at the bottom, clip to see your slide, and there's a diaphragm at the bottom, which allows the light to enter the field of view. Then there's a light source at the bottom, at the base, where it's a base which supports the microscope. So together it makes your microscope. Ocular lens, body tube, nose piece, objective, stage clips, diaphragm, light, arm, stage, coarse adjustments, fine adjustments, and base. So all together is your microscope. So in the field of view, whatever you are seeing, you're seeing upside down, um, and also opposite of it. That's not the straight. And you can have the magnification from 7x to 10x as per your objective lens. If your image of a teratoma, some carcinoma cell lines look like this, though in that higher magnifications you can go up to like that. And then to calculate the power of magnification, multiply the power of ocular lens by the power of the objective. For example, if it is 10x, here is a 40x. Then the magnification will be multiply the eyepiece power with the objective power. So if it is 10x is the eyepiece, objective is 100x. So your magnification will be 1000x. Like this. And object that you see in the microscope, they're upside down and backward, and movement appears to be in opposite direction than the actual movement. That slides to move. Then if you move your slide from left to right or right to left, you will see things like this in that. So that's it. So recording observations. This is what we used to do in the lab, but not here. And while making a wet mount lab, clean your slide with a tweezer to lay the specimens on the drop of water and gently touch the cover slip to the edge of the drop of water to cover the specimen of the water. You drop one drop of water or the oil in the middle. Then put your cover slip at 45 degrees Celsius on the top. Then slowly you lower it slowly at the on the top of your sample, so that you should not have any air bubble to that. So that's it. Very nutshell, simple. Um, Nothing so hard and fast in that. Microscope basics. Now we go to a bit advanced version of it. If you have any questions, students, feel free to write in there. I will happy to reply to you. Only 14 students. Come on. So we have in total how many? Around 40 or 35, I can say. You are around 35. And half of the students are coming. That's very sad. So let's start with light and electron microscopy. So this is the replica that you are seeing here of a Leeuwenhoek microscope from 1670s and medical library from the medical branch. So this is one another replica from the Culpeper tripod microscope, which was built in 1725 by Edmund Culpeper and the collection of Moody Medical Library. So this is how the old time the microscopes looks like. So for today, the content for this microscopy will be dealing with light microscopy, electron microscopy, and specimen preparation. So in the types of microscopy within the light microscopy, we will deal with the 7 types of microscopy. That is bright field, dark field, phase contrast, polarizing, DIC, epifluorescence, confocal laser scanning. Where in the electron microscopy, we will have scanning, transmission, then further specimen preparations, light microscopy, scanning electron microscopy, and transmission electron microscopy. So all these parts include in it. So let's start. So basic idea that I showed you before, the very basics of a microscopy. The reason behind that was mainly due to to get you familiar with the microscopy world. Only then might be we are able to understand things. So microscopy have been very essential tools for cell biologists. This PPT will basically provide you how you can prepare your biological studies, general techniques, and how can you use your light microscopy and electron microscopy in order to see the world of cellular biology. So first thing first, bright field microscopy, the basic microscopy, is the most common general use microscope. Bright field microscopes are named because the microscope field is bright while the object being viewed is dark in that. So simple design, light directed at specimen absorbed to form image, and then unstained specimens have poor contrast, whereas a stained specimen shows excellent contrast in that. So ideal for stained bacteria, cells, and tissues. So pretty good in that case. So this is the binocular bright field microscope with movable stage. Stage could be moved, and iris diaphragm built-in light source. So we use them for the clinical research as well as student microscope from day-to-day life. Then comes your bright field image. How the image looks like is a section of gut tissues containing ciliated parasites. So you can see some ciliated parasites of gut tissues being affected by parasites here. Flagellates, trichomonas, stained blood cells in peripheral blood smear. So all these things belong to bright field image here. Then there are some inverted microscope, which is a modification of bright field microscope, in which the light source is from the top and objective lens is at the bottom. So inverted microscope at the positions of light source objective is inverted and light source is above specimen, and they are located beneath the stage. Then dark field microscope, here we can see that are the one which creates a dark background to allow the viewing of the small unstained objects such as motile bacteria that would be difficult to view in the bright field. So the central portion of the light is blocked so that only oblique light strike the specimens, scattering light rays then that enter the object from the image. So this is from the 19th century. We use bright specimens, dark background in this. Light is not scattered by the specimens, therefore making the field dark. We can see very small objects, but resolution is variable, and high contrast, good for unstained live and motile specimens. Then this is the dark field image here. On this note, the alga hydrodictone reticulum viewed with a dark field microscopy by Dr. D. Folkretz from AU. So here you can see very nicely this algae network is being shown in the dark field. So you can see the background being dark in this side, so that your images are pretty much good. This is the Leptospira spirochete viewed with the dark field microscope here. So it's better than bright field microscope. Then comes your phase contrast microscope, which converts the differences in refractive index in specimens to differences in image brightness. The central portion of the light source is blocked, creating a ring of light from the condenser that illuminates the specimen. And the light waves refracted by the specimens are slowed by a phase retardation in that case, increasing the difference in the wavelength between refractive and unrefractive rays. So when the refractive and unrefracted waves are focused, they produce an interference due to the difference in the wavelength, and this is seen as a difference in the brightness in the specimen. So this is the technology from 1940s. Provides high contrast, good resolutions, good for bacteria, flagella, cilia, organelles such as mitochondria, and good for unstained or live mounts and artifacts also. So this is the images of phase contrast microscope on the left as compared with the DIC image here. The phase contrast has better, this is the epithelial cells, squamous epithelial cells. Shows the cells to be particular. You can see them cell organelles can't be seen very well in this. We can see them as small dots, but we can see precisely the nucleus, the cell membrane, the cell wall around it, but the cell structural organelles are not so beautifully could be seen in this part. Then comes polarization microscope, which detects specimens that are birefringent, that is have a characteristics of a double diffraction, that is velocity of light of refraction substance is not the same in all directions. So in this we use a polarizer of like there are two polarizers and in between your specimens, and both are 90 degrees parallel to it, and we have a bright and dark image out of it, and we use it is used for the molecular structure such as crystals, minerals can be quantified in this part. So this is some minerals, meteoroid, Martian meteoroid with 0.3 millimeter depth viewed under the polarizing microscope. On the right hand side we can see renal epithelial cells containing fat globules, and bottom right is the high magnification view of same cells viewed with the polarizing microscope showing typical Maltese cross. So in this way we have three different microscopes. So epifluorescence microscopy, which allows the detection of molecules and ions within the cells, and fluorescent dyes absorb short wavelength of light and emit longer wavelengths. So we use UV as a light source in that. Mercury is known as arc, high contrast, high resolution image. Special fluorescent dyes used to locate molecules in a specimen. Black background, bright stained specimens, no condenser required, and light comes from above the specimen. And we detect small quantities molecules, can be used antibody staining techniques in this case. Further, the epifluorescent images looks like this. So on the left hand side you can see fluorescein isocyanate which emits green light in the tetrahymena ciliated protozoan, and on the other side, on the right hand side, the cell is stained with the antibody against cell membrane surface antigens which are used for the fluorescent technique with bright field of tetrahymena, and this is the bright field, and these are the epifluorescent images of the same. So things are pretty much bright and it's pretty much nicely visible. And here you can see some epifluorescent images showing the metaphase with the help of this microscope in the lung cells. So here you can see very nice tubulin, keratin, DNA parts to be separated. Blue is the metaphase chromosomes being separated here, the DNA, and the red is the keratin filaments, where the yellow is the spindle apparatus mode of microtubules. And this is images from the New York State Department Health, Albany, New York. From there these images were created. So very nice image where you can see beautiful the combination of red, yellow, blue, how your during your metaphase state of your meiosis, or it is mitosis, I don't know which state it is, with the microtubules on at the background. So everything has been depicted very nicely and beautifully with this help of microscope. Then comes your further differential interference contrast microscope. I'm telling you these different types of microscope, but because when you will be in the lab, whenever you will start working in the lab professionally, um, it depending upon the kind of lab you are, there might be different kind of microscope, and in order to find the best results for your paper or for your research, you have to identify which microscope will be best for your results. Either go for epifluorescence, either go for phase contrast, either go for polarizing microscope, either go for simply bright field or dark field, um, or you can go for the advanced electron microscope. Yeah, so there are many options, many things are available: scanning electron microscope, transmission electron microscope, like in this case differential interference contrast, and this is done to check the 3D images, which gives quite high resolutions, high numerical aperture, high contrast. It has a mercury lamp on the side with a beam splitter, prism, polarizing filter. So it has a combination of all the microscope. This is a quite advanced one, this TIC. So this is how you see the images, like quite 3D images in this. So this is of the cricket, you know, this which make this sounds in the park. Chi 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 chi. These are the oocytes are being shown here, and the Balbany bodies are being shown also, like on the small one. So this is the best way to see the I mean this is actually this the B1 is the Balbany bodies. Here we can see, yeah, this is from the fluorescent stains with Balbany bodies. We are seeing it. Then comes your main confocal laser scanning microscope. This is the image that I'm showing is pretty old, but now very advanced versions have been there in the labs, and they are quite expensive. We can't even think in our minds. They are pretty expensive, but whosoever these facilities are, you can do really good science with that. You can have really good 3D images. Even I got some in my PhD. So they have very high resolution, sharp image, quite sensitive, and we use argon or krypton laser here to see your image, and we eliminate all the background, all the interference, and it's one of the best one, if you can get it. So these are some confocal images that you can get from it. So So the left image is showing you optical sections of trichomonas immunostained with antitubulin and fluorescent. This fits that we use mostly antibody, quite common in the lab. And the green structure consists of microtubules here. So here we have PF posterior flagellum, AF is the anterior flagellum, C is the costa, and exostyle. This is the red arrow. On the right hand side is a bright field image of the same. Here you have posterior flagellum of trichomonas, and can be seen, but exostyle red arrow is unstained. We can't see this exostyle, this double layer, but this is not present here. So you can see the how difference it is as compared to your bright field and the confocal image. कह सकते हैं ज़मीन आसमान का फ़र्क है इसके अंदर. There's a lot of difference in that. Yeah. So then next is electron microscopy, which is your scanning electron microscopy, transmission electron microscopy. So scanning electron microscopy includes your fixed dehydrated specimens which are mounted on the stubs and surface coated with the gold, palladium and rhodium, and the specimen is placed in vacuum and electron beam scans back and forth over it, and electrons that bounce off the metal coated specimens surface are collected, and you can convert this digital image displayed on TV like monitor, and this provides also a 3D image. The scanning electron microscope quite high resolutions, and then it's a focused using a magnetic field. So SEM images, they are the comparisons of SEM images with images obtained using your DIC microscope. So DIC microscope and SEM reveal specimen surface topography. Much greater detail is obtained using SEM because of greater resolution and magnifications capability. So on the top left you can see the trichomonas. On the left side you can see how 3D the things are. Earlier we were seeing most of the images to be 2D, but in this we can see things to be more particular and more compact and very nicely. Further, transmission electron microscopy, which fixed dehydrated specimens are embedded in a resin's hardened section stained with heavy metals such as uranium, lead, and inserted into the electron column. So it's a 2D image, reveals internal cell structure, high resolutions, high magnifications, electron beam is focused by the magnetic field. And here you can see comparisons of a TEM and SEM. So on the left hand side is a SEM and on the right hand side is a TEM. So we can see straightforwardly that the organelles of the TEM, they are more profound and more better, and we can see them each structure to be very nicely be shown here, whereas in the SEM we can see the structure, but it's good for the image forming but not good enough to see the internal organelles. And these are further some more TM, your transmission electron microscope of rat liver cells on the left side and the pancreas acinar cells on the right hand side, which revealing the internal cell structure. So you have nucleus, you have a nucleolus, rough endoplasmic reticulum, and CB, cell boundary, zymogen granules, and mitochondria. So this is the specimen preparation with the light microscopy, scanning electron microscopy, and transmission electron microscopy. How you can prepare your specimens in that. So for the general schematic for preparing specimens for light microscopy, you have your live mounts here. You can see the view. Then there's vital stain, then you can view. Then to preserve them, to fix them for specimens for histology. First you fix, then remove the water, then embed them, section them, mount them, stain them, and then you can view them. So live mounts are the way of seeing your organisms in the low contrast in order to see your things pretty good. So fixed specimens for histology, the study of tissue. Specimens may be preserved using chemicals. We can use some chemicals like formalin, acetic acids, ethanol and methanol in order to fix your proteins and lipids. And fixed specimens are dehydrated by serial transfer through an ascending alcohol series to 100% alcohol. And specimens are infiltrated with melted paraffin, paraffin substitutes, or plastic and placed in mold to harden it. Then specimens are cut into 5 into 10 micromolar thick sections using steel knife or razor on an instrument called a microtome. So here you can use vibrotome using vibrating knife to cut the sections. And sections are then mounted on slides, stained to achieve contrast or identify cell structures or components, and viewed microscopically. So many variations in techniques are used to prepare specimens for light microscopy. Some omit the dehydration, infiltrations, embedding, sectioning steps, and use of aqueous staining systems for viewing whole mounts. So freezing has to be done with instead of chemicals to fix tissues that need to be examined. And examples are frozen biopsies, tissues in which heat liable structures are to be stained. And frozen specimens are also sectioned using cryotome, a microtome in case freezing chamber. So for the scanning electron microscope, to prepare your specimens, you fix, dehydrate, mount on the stubs, sputter coat, and observe them. So to do so, first fixation is done with the help of glutaraldehyde, paraformaldehyde, osmium tetroxide. Then you dehydrate them by using sanding alcohol series from 100% to the organic solvents such as acetone, propylene oxide. Specimen for SEM may also be processed in critical point drying apparatus. So specimens are mounted on aluminium stubs using sticky tapes here. So example of one of the type of stub is this. So we put your sample on this powder coater, which are coated with the gold, palladium, or rhodium, quite expensive. You can see these stubs quite expensive. In a special chamber to cover the specimens with 10 to 20 millimeter thick metal layer. So this is how the stub is inserted into the SEM scanning electron microscope scan and then observed over the video. So it's quite professional. For a transmission electron microscope, you fix, dehydrate, then infiltrate, and then section, apply sections to grids, stains, and then you observe it. So fixation specimens are fixed in glutaraldehyde or formaldehyde or glutaraldehyde mixtures, followed by osmium tetroxide. Then dehydration is accomplished by carrying the specimens through an ascending alcohol series to 100% alcohol, then to organic solvents such as acetone or propylene oxide. Specimens are then infiltrated with an epoxy, plastic resins, and placed in plastic molds to harden it. So as an instrument, this is called the ultra microtome. Here you can use your to section the specimens. So most of the histological labs, if you entered, you can see these knife holders there and specimens holders there. So both works nicely in order to work. Then glass or diamond knives are used to cut the ultra thin sections. So you can see your sections being cutted with ultra thin sections of 1 millimeter wide, and you can put them over your slide, glass slide, and fix them, and then do your homotaxin HE staining, yeah. Then you can watch them under your microscope. So I did this for 1 year almost as a technician in the lab of Max Planck Institute. So quite boring work, but a lot of interesting data is coming out. Then these are sections transferred to tiny metal grids for sport. This function of the glass lid in LM. So this is a close view of a one type of grid. So you can see so how tiny is your grid is of a forceps. So heavy metal stains such as uranyl acetate, lead citrate are applied to make certain structures electron dense in that case. So these are the grids are then inserted into the transmission electron microscope, and then you observe them. So this is how big your TEM looks like. So that's it for today, I guess. Uh, so I will share this part. Light and electron microscope. HE staining. So, bache, basically, this is done to check your tissues, of biopsy tissues, of cancerous tissues, using the homotoxylin nuclear stains, in order to check squamous part, how things are working. Maybe I can show you one video regarding that. Homotoxin solution. I hope your query is clear. You're welcome. So I can't start today the next topic because if I start that, it will be left in between. So we will stop it here. That's it for today. We see us tomorrow, same time, same place. Thank you very much.